Parasympathetic system. The parasympathetic system can be divided into cranial and sacral according to the location of its nuclei. Cranial parasympathetic system. The visual effector nuclei of the cranial division are part of the oculomotor, facial, glossopharyngeal, vagus and accessory nerves. The oculomotor nerve parasympathetic system is made up of nerve fibers that are involved in the motor innervation of the ciliary muscle and the sphincter muscle of the eye pupil. Preganglionic fibers stem from the Edinger and Westphal nucleus, which we examined in the section dedicated to the oculomotor nerve, and they travel towards the ciliary ganglion following the path of the inferior branch of this nerve. The ciliary ganglion, located within the orbital cavity next to the optic nerve, is the connecting point not only for preganglionic fibers of the parasympathetic system, but also of sensory and orthosympathetic fibers. It consists of three roots: short, long, and orthosympathetic. The parasympathetic fibers originating from the Edinger and Westphal nucleus make up the short root. The long root consists of sensory fibers that originate in the nasal ciliary nerve, which is the branch of the ophthalmic nerve, a division of the trigeminal nerve. They are responsible for eye sensitivity. The orthosympathetic root stems from the internal carotid plexus of the orthosympathetic nervous system. These fibers play a vasomotor function, thus vasodilation and vasoconstriction, and a motor function on the pupil dilator muscle. It's worth mentioning that sensory and orthosympathetic fibers pass through the ganglion without interruption. Therefore, the ganglion is only a sort of gateway for these types of fibers to reach the eye. Originating from the ciliary ganglion, postganglionic fibers make up short ciliary nerves which head to the eye. In this case, the parasympathetic system will innervate the iris sphincter muscle, thereby controlling its narrowing, and therefore pupil restriction, miosis, in response to light and visual accommodation. Meanwhile, the orthosympathetic fibers innervate the iris dilator muscle, leading to pupil dilation, a process known as myidraiasis. It's no coincidence then that people's pupils dilate when they are afraid. It's the effect of the orthosympathetic system. The so-called fight or flight response we've mentioned before, that once triggered controls each of these diversified reactions. The facial nerve parasympathetic system can be divided into two components, one belonging to the proper facial nerve and the other to the intermediate nerve. The proper facial nerve parasympathetic system. The proper facial nerve parasympathetic enables the production of tears by the lacrimal gland and saliva by the palatine glands. Overall, this function is referred to as excitosecretory because it involves gland stimulation to produce their secretions. The preganglionic fibers originate in the lacrimal nucleus located in the caudal pons. These fibers join the proper facial nerve and then the greater petrosal nerve. Then they merge with the fibers stemming from the deep petrosal nerve, which belongs to the orthosympathetic system. The union of the parasympathetic and orthosympathetic fibers result in the pterygoid canal nerve, or Vidian nerve, which reaches the pterygopalatine ganglion. The pterygopalatine ganglion located in the pterygopalatine fossa, receives not only the parasympathetic and sympathetic fibers through the Vidian nerve, but also sensory fibers from the maxillary division of the trigeminal nerve. Therefore, in the same way as observed with the oculomotor nerve parasympathetic system, the sympathetic and sensory fibers pass through the ganglion without synapsing, where the parasympathetic fibers synapse with the ganglion itself. A particularly interesting connection is with the maxillary nerve. Not only does the maxillary nerve release preganglionic fibers directed to the sphenopalatine ganglion, but it also receives postganglionic fibers from the latter. These fibers, following the path of the maxillary nerve, enter the zygomatic nerve and then, through anastomosis with the lacrimal nerve, travel along with it to the lacrimal gland, thereby innervating it. But now let's look at the efferent branches, that is, those that originate in the sphenopalatine ganglion. The efferent branches include posterior, superior nasal nerves and palating nerves. Posterior superior nasal nerves contain sensory fibers stemming from the maxillary nerve and postganglionic fibers, originating in the sphenopalatine ganglion. Sensory fibers innervate the mucosa of the navel fossa and that of the anterior part of the hard palate, while the postganglionic fibers innervate the glands located in these mucous membranes. Palatine nerves are divided into three classes: anterior, middle, and posterior. These three nerves all contain sensory fibers stemming from the maxillary nerve for the innervation of the mucosa of the hard palate and soft palate, and postganglionic fibers for the innervation of the associated glands. The immediate nerve parasympathetic system. The immediate nerve parasympathetic system allows for the excitosecretory innervation of the submandibular, sublingual, and lingual glands. Preganglionic fibers originate in the superior salivary nucleus located in the caudal pons. From there, after entering the immediate nerve, they pass into the cauda tympani nerve and then into the lingual nerve, a branch of the mandibular division of the trigeminal nerve. This passage of fibers is made possible by the anastomosis between the cauda tympani and the lingual nerve. From the lingual nerve, fibers head to two different ganglia, the submandibular and the sublingual ganglion. The submandibular and sublingual ganglia, located respectively in the submandibular and sublingual fossae, receive only the preganglionic parasympathetic fibers by the lingual nerve. The resulting postganglionic fibers head to the submandibular and sublingual glands, as well as the salivary glands of the tongue, thereby controlling their secretion. The Glossopharyngeal parasympathetic system includes a set of visceral efferent fibers and a set of visceral sensory fibers. The visceral efferent fibers enables the excitosecretory innervation of the parotid gland and of the molar and labial salivary glands, that is, those located in the mucous membrane of the cheeks and lips. The preganglionic fibers stem from the inferior salivary nucleus within the medulla oblongata. Their pathway follows the glossopharyngeal nerve, then the tympanic nerve, and lastly the superficial small petrous nerve, until reaching the otic ganglion. The otic ganglion of Arnold, located in the infratemporal or zygomatic fossa, only receives the parasympathetic fibers of the glossopharyngeal nerve, but has different postganglionic fibers. Most postganglionic fibers enter the auriculotemporal nerve, a branch of mandibular nerve of the trigeminal nerve. Following the pathway of this nerve, they reach the parotid gland, enabling its secretion. Other postganglionic fibers pass through the mandibular nerve itself and then enter the buccal nerve and reach the glands located at the level of the lips and cheeks. Instead, visual sensory fibers convey taste and general sensation of the posterior third of the tongue and palate. These fibers originate in the petrous ganglion and their central extensions end in the nucleus of the solitary tract, while the peripheral extensions travel through the pharyngeal branches, the lingual nerve and the carotid sinus nerve to convey stimuli arising from changes in blood pH. The vagus nerve parasympathetic system is the most widespread part of the cranial parasympathetic system because its visual efferent fibers enable the innervation of the involuntary muscles and of many glands within the body. Preganglionic fibers stem from the dorsal motor nucleus of the vagus nerve located in the medulla oblongata. They follow the branches originated in the vagus nerve itself, in the thorax and abdomen, all the way to the various parasympathetic ganglia. There are several ganglia of the vagus nerve located close to or within the mucous membranes of the organs they innervate. Postganglionic fibers allow for a broad innervation. They follow basically all branches stemming from the dorsal motor nucleus, which we already examined when discussing the vagus nerve. Therefore, we will only make a list of the innervated organs: heart, trachea, together with its glands, bronchial glands, the entire digestive system, including the attached glands, that is, liver and pancreas, but also those distributed throughout the digestive tract, urinary tract, and spleen. Again, there are visual sensory fibers that originate in the inferior ganglion of the vagus nerve, also called the nodus ganglion, and reach the nucleus of the solitary tract and the peripheral cardiovascular areas. The accessory nerve parasympathetic system is deeply linked to the vagus one. Preganglionic fibers stem from the accessory nucleus of the vagus nerve. From there, they follow the accessory nerve itself and then join the vagus nerve. Lastly, they follow its pathway. The sacral part of the parasympathetic system involves visual effector nuclei within the spinal cord, in the sacral area. There are two columns of neurons, one lateral and one medial, located at the bases of the anterior horn of grey matter. Clearly, preganglionic fibers originate from the two columns, thereby following the anterior roots of the sacral nerves to reach the pelvic ganglia. Parasympathetic pelvic ganglia are scattered within the pelvic plexus, which, as we saw in the last video, belongs to the orthosympathetic system. Originating from these ganglia, postganglionic fibers enter the pelvic plexus itself and then spread to various organs: rectum, the last tract of the ureter, bladder, urethra. In males also, prostrate, seminal vesicles, ductus deferens, corpora cavernosa of the penis, while in females, uterus, vagina, corpora cavernosa of the clitoris, and vestibular bulb.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.